Hello everyone. Today we will solve some important questions on cell signaling for CSA NET, ICMA NET, GET and various other exams. This topic is very important. So stay tuned. First question. Which of the following are the basic categories of chemical signaling found in multicellular organisms? Option 1 autocrine signaling, option 2 paracrine signaling, option 3 endocrine signaling, option 4 all of the above. And the correct answer is option 4, all of the above. Now, these cells communicate via various types of signaling that allow chemicals to travel to target sites in order to elicit a response. There are four basic categories of chemical signaling which are found in multicellular organisms. These are endocrine signaling, autocrine signaling, paracrine signaling, and signaling by direct contact. In endocrine signaling, the signals from distant cells that originate from the endocrine cells, usually producing a slow response, but having a long-lasting effect. Second is autocrine signaling. It is produced by signaling cells that can also bind to the ligand that is released. The signaling cells and the target cell can be the same or the similar cells. The auto means selves. Third is paracrine signaling. The paracrine signaling is from a cell signaling in which the target cell is near the signal releasing cells. So the para means near. So here the target cell is near the signal releasing cells. Number 4 is signaling by direct contact. It occurs by transferring signaling molecules across gap junctions between neighboring cells. Next question. The hormone, also called the ligand, is considered as option 1 first messenger, option 2 second messenger, option 3 both 1 and 2, and the option 4 none of the above. And the correct answer is option 1 first messenger. Now, why hormone are called as first messenger? The hormone are referred as first messengers because once bound to their receptor, they initiate the associated biological response. For peptide hormones, this response is then amplified via a second messenger system. However, it is binding of the hormone of the receptor that initiate the response. There are signaling molecules and are synthesized within the body that regulate and coordinate physiological and metabolic functions by acting on receptors located on or in the target cells. They can be secreted by specific secretory cells that are either localized in secretory organs. Next question. In which type of signaling the cell that express messenger molecules also produces receptor? Option 1 autocrine, option 2 paracrine, option 3 endocrine, option 4 all of the above. We have already discussed this. The answer will be option 1 autocrine. In autocrine signaling, the cells that expresses messenger molecules also synthesize the surface receptor. The surface receptor can respond to the messenger synthesized by the cell itself. The auto means self we have already discussed. Next, paracrine messenger molecules are usually option 1 large, option 2 stable, option 3 unstable, option 4 amphoteric. And the correct answer is option 3 unstable. Why these are unstable? The paracrine messenger molecules are unstable and cannot travel long distance in the body, mostly because they are either degraded by the enzymes or they bind to the extracellular matrix. That's why they are unstable. Next question. The largest family of human cell surface receptors is the option 1 receptor tyrosine kinase, option 2 G protein coupled receptors, option 3 enzyme linked receptor, option 4 ion channel linked receptors. And the correct answer is option 2 G protein coupled receptors. It is also known as 7-pass transmembrane domain receptor, STM receptors, hepatohelical receptor, serpentine receptors, and G protein linked receptors. The two principal signal transduction pathways involving the G protein coupled receptors are one is CAMP signal pathway and the second is phosphatidylinositol signal pathway. Next question. Which of the following ligands binds to the extracellular receptor? Option 1 nitric oxide, option 2 thyroid hormone, option 3 steroid hormone, option 4 is growth hormone. And the correct answer is option 4 growth hormone. So growth hormone binds to the extracellular receptors, whereas the thyroid hormone and steroid hormone interact with the intracellular receptors. The steroid hormone and thyroid hormone interact with intracellular receptors, that is either the nucleus or the cytoplasm. Okay. Whereas the protein hormones, that is and protein hormones such as the growth hormone, prostaglandins and catecholamines interact with the extracellular ligand binding domains of the plasma membrane spanning receptors. That's why the growth hormone binds the extracellular receptors. So this answer is correct. Okay. Next question. Specialized receptors in the vertebrate eye that responds to light signals are option 1 phytochrome, option 2 rhodopsin, option 3 melanophores, option 4 cryptochrome. And the correct answer is option 2 rhodopsin. Rhodopsins is a member of a class A of the GPCR superfamily, that is G protein coupled receptor family, which is a large group of cell surface signaling receptors that transduce signals into intracellular pathway through the activation of heterotrimeric G protein. In rhodopsin signaling pathway, light is converted to an electrical signal in the retina of the eye. Next question. Retrograde signaling involves communication of option 1 nucleus to the chloroplast, option 2 mitochondria to the nucleus, option 3 nucleus to the mitochondria, option 4 chloroplast to the mitochondria. And the correct answer is option 2 mitochondria to the nucleus. Now what is this retrograde signaling? Retrograde signaling conceptually means the transfer of signals from the semi-autonomous cell organelles like mitochondrion or the chloroplast to the nucleus to modulate nuclear gene expression. The retrograde signaling pathway in biology is the process where signal travels backward from a target to source to its original source. For example, the nucleus of a cell is the original source for creating signaling proteins. During retrograde signaling, instead of signals leaving the nucleus, they are sent to the nucleus. Next question. Which of the following is a cell surface receptor? Option 1 enzyme linked receptor, option 2 G protein coupled receptor, option 3 ion channel linked receptor, option 4 all of the above. And the correct answer is all of the above. That is enzyme linked receptor, G protein linked receptors, ion channel linked receptors, all are cell surface receptors. We have already discussed about G protein linked receptors. Now let's discuss about enzyme linked receptors and ion channel linked receptors. The ion channel linked receptors are usually multimeric protein which is located in the plasma membrane. Each of these proteins arrange itself so that it forms a passageway or pore extending from one side of the membrane to the other to allow ion to pass through. Next is enzyme linked receptors. It is also known as the catalytic receptors. These are proteins that act both as receptor and enzymes. They are transmembrane proteins that have ligand binding site on the outside of the cell and an enzyme activity or association on the inside. So these enzyme linked receptors, ion channel receptors and G protein linked receptors all are cell surface receptors. Next question. Cholera toxin causes massive and often fatal diarrhea by option 1 inactivating G1 protein, option 2 irreversibly activating adenylate cyclase, option 3 locking GS protein into an active form, option 4 rapidly hydrolyzing G protein GTP into GTP. And the correct answer is option 2 irreversibly activating adenylate cyclase. Cholera toxin causes massive diarrhea by activating adenylate cyclase CAMP system which is located at the basolateral membrane of the intestinal epithelial cells. As it activates the adenylate cyclase system, it increases the production of CAMPs, that is cyclic AMP. The increased cyclic AMP levels mainly responsible for the altered transport of sodium ion and chloride ion, which causes the massive and fatal diarrhea. Next question. Steroid, a biologically active organic compound, is derived from option 1 hormone, option 2 chemicals, option 3 cholesterol, option 4 carbohydrate. And the correct answer is option 1 hormone. The steroid hormone regulates cellular process by binding to intracellular receptors. We have already discussed that steroid hormone, thyroid, all are intracellular receptors that in turn interact with discrete nucleotide sequences to alter the gene expression. Because most steroid receptors in target cells are located in the cytoplasm, they need to get into the nucleus to alter the gene expression. Next question. Which of the following has the highest affinity of ligand-receptor interaction? Option 1 insulin receptor, option 2 lectin sugar receptor, option 3 biotin avidin, option 4 cytokine receptor. And the correct answer is option 3 biotin avidin. Avidin is a tetrameric glycoprotein. Each of its 4 monomer can bind biotin. Avidin binds biotin very tightly and specifically. The interaction between avidin and biotin is one of the strongest non-covalent interaction in nature. So, the biotin-avidin has the highest affinity ligand-receptor interaction. Thank you. If you like this video, please like, share and subscribe to this channel. Thank you.